Hi everyone, this is Professor Howard. In this lecture, I'm here with my son George. George, do you want to say hi? We're here to talk about synaptic transmission, or the communication between one presynaptic neuron and a postsynaptic neuron. I'm going to have a big emphasis on neurotransmitter vesicles and an emphasis on the life cycle of a neurotransmitter vesicles. I'll also contrast chemical synapses with electrical synapses. Let's get started. Okay, again, as always, take a look at these major concepts to master. If you can address all these questions, then you have a pretty good understanding of the material. If you have need for additional resources to help you understand some of this, take a look at this chapter, which I'm going to post in the folder on Blackboard. Okay, there are two types of synapses in the brain. There are chemical synapses and electrical synapses. Chemical synapses release neurotransmitters into a wide synaptic cleft where they can freely diffuse across this distance and bind to postsynaptic receptors. To contrast that, electrical synapses are quite a bit faster. There are direct ionic channels allowing the flow of ions between the presynaptic and postsynaptic element. What's interesting about these, an action potential will arrive here, and you will see an excitatory postsynaptic potential occur in sort of a delayed manner in a chemical synapse. In an electrical synapse, the shape of whatever occurs here is going to be mirrored postsynaptically, but it will be degraded. Okay, so just to reiterate that point, imagine if we had two neurons that are connected via these electrical synapses. If you were to pump some sort of current into this presynaptic neuron here, and let's say we just inject this square pulse of current into the cell, you might expect to see this current diffuse via all of these properties we've mentioned, so including having a time constant and to be graded in some capacity, so you might expect this to actually go down. As this current spills into the next cell where it's connected electrically, you see that that signal is further degraded, suggesting that it gets lost through the membrane along the way. But the shape of this reflects pretty directly the shape of what's happening presynaptically. This is an extremely fast process, so you see that the amount of time between here and here is almost zero. And again, the shape is mimicked. So in an exam setting, if I ask you, how could you differentiate between electrical and chemical synapses, that might be a good way to do that. So what's actually happening at these connections? Connections in an electrical synapse are comprised of these molecules known as connexin. This is a hemichannel, and what that means is each of these channels has two major components to it. They sit sort of connected on top of one another. Each is expressed in the presynaptic and the postsynaptic element, and where they bind is what makes it a functional channel. Each of these is also comprised of 6 subunits that is repeated. You need both halves to form a channel. They sit outside to outside, and that's what allows the passage of ions between these different neurons. Interestingly, this is not just a sort of passive phenomenon. Connexin channels, when they're in the presence of high calcium, will close and shut down. And that's one way by which these cells can regulate how much activity they're passing between each other. Another point to point out here is that this synaptic cleft in an electrical synapse, because it's restricted based on protein-protein interactions, this connection is going to be very small and much smaller than what you'll find in a chemical synapse. Here's an actual scanning electron micrograph of one of these connexin-based connections between these electrical synapses. Okay, and why electrical synapses? What's the purpose of this? You often find electrical synapses where you find synchronized neuronal activity. So if you have multiple neurons that you want activated all at the same time, then it's a good idea to have them linked via some electrical synapse. They're best known for their functionality in defensive or escape circuits, because you need to enact these types of behaviors extremely quickly. You don't have time to sit around and wait for chemical synapses to take over. So this is the deep sea slug aplesia. We'll see aplesia again later in class, because a lot of synaptic plasticity was characterized using this as a model of a simple synapse. If you stimulate the animal's tail, you see that this sensory neuron is linked to these motor neurons that cause this ink gland to be constricted, and the animal will then defensively release ink. Because you want this to happen evenly at all these different places, it's synchronized via these electrical synapses. So if we were to record across each of these neurons, you see that the activity is totally linked. Give me 1 second. What's up, buddy? George got the candy bowl. Okay. We'll change gears a little bit next. We're next going to talk a little bit about chemical synapses. The benefit of a chemical synapse is a signal gets amplified when you have a chemical synapse. That's because this can generate a downstream action potential. The other thing to note is that any G-coupled protein receptor or G-coupled postsynaptic receptor can cause widespread changes in cell activity. So this is going to be a much more complex and potent response relative to an electrical synapse. These are definitely more common in both invertebrate and vertebrate nervous systems. And here is a nice scanning electron micrograph showing, these are mitochondria here. These are synaptic vesicles which store the neurotransmitter. These are the active zones here where we have neurotransmitter binding and postsynaptic receptors. Neurotransmitter release is dependent on calcium, intracellular calcium, which is typically very low in concentration. When an action potential arrives at a presynaptic terminal, so here you can see an action potential arriving at this presynaptic terminal, you get the influx of calcium. But this is in a delayed manner, and that's because during this depolarization phase, even though the calcium channels are open, calcium doesn't flow in because you're so close to the reversal potential for calcium. So here you can see the IV curve for a typical calcium channel, and somewhere around 0 volts or so, you do get a very potent influx of calcium. But at higher voltages, that's not the case. So what's happening at these higher voltages is calcium is delayed in its entry, and that delay is actually the longest period of time between the presynaptic action potential and a postsynaptic EPSP. So this is a major factor in terms of the amount of time it takes for a chemical synapse, which is inherently slower than an electrical synapse. Another important point here is that it's this dropping of the voltage that leads to a massive influx in calcium in these cells. So here is a series of voltage clamp recordings, and you see that there's a little bit of calcium influx, but calcium notoriously has this large tail at the end of these signals. And that's because as the voltage falls in these voltage clamp recordings, this is allowing a stronger driving force on calcium to enter the cell. So just before the actual channel shuts down, you get a nice big spike in calcium. So the cell can regulate total numbers of calcium, or total amount of calcium, and very quickly shut off the supply. Okay, so I just want to provide a quick overview of what happens at the presynaptic terminal prior to neurotransmitter release. Initially, we have an action potential in the presynaptic element. This leads to a PSP, or an EPSP, excitatory postsynaptic potential in the postsynaptic cell or element. So what happens in between is we have depolarization, the opening of voltage-gated calcium channels, the binding of vesicles to the membrane, the release of neurotransmitter into the synaptic cleft, the binding of neurotransmitter onto these are ionotropic receptors, and the influx of sodium depolarizing the postsynaptic element. Yeah, note here there's a long delay between this dotted line where the action potential is and the actual EPSP. And again, the mechanism here is the delayed influx of calcium, despite that channel being open. Now what I'm going to do is walk you through this step by step and go into a little bit more detail in each one of these phases. So this is going to be a detailed view of how a synaptic vesicle comes into existence and then how it does its work. First of all, parts of the vesicle, including that structure I showed on the very first slide of this lecture, these are spheres that are dotted with all manner of different types of proteins. So all of these proteins and the components, including the cell membrane, they all have to be made somewhere in the cell. They're actually synthesized in the cell body, and they get transported down these microtubule highways through the axons of these cells. This is via fast axonal transport, which includes the walking kinesin molecule. You don't have to know that, but just know that these parts are first made in the cell body and then transported down to the cell terminal. Vesicles are initially attached to actin fibers in the so-called reserve pool, where they will remain until they are needed or recruited during periods of high activity. And in synaptic plasticity, which we're going to talk about in the future, this is one of the key mechanisms of very fast synaptic plasticity, or changes in the synaptic strength between connected neurons. Vesicles also can migrate down and join this so-called readily releasable pool, which is found more downstream, closer to the actual active site of these synapses. That's where they're going to sit and wait to be released into the synaptic cleft. Once they're sitting there, they're awaiting the arrival of an action potential where these voltage-gated calcium channels, these are N-type channels, are going to open. But neurotransmitter vesicle binding is only going to occur in very, very high concentrations of calcium. So what happens here is you get these little so-called microdomains where this is a voltage-gated channel here, this little dark circle, and this area immediately around it is an area of very high calcium concentration. So extracellularly, we have very high calcium concentration, very low concentration of calcium inside of the cell. And in this area, before diffusion makes this spread evenly, we have really high concentrations as soon as those voltage-gated channels open. So these little microdomains, if you sort of took a cross-section of a single plane of this surface here, you'd see these areas where calcium levels are really, really high. And most neurotransmitter binding and release into the synaptic cleft actually happens between a couple of overlapping microdomains, which would be shown here as these areas with 2 spikes in them instead of just 1. The reason for that, again, is because calcium levels need to be high before you get binding and fusion of the vesicle with the membrane. Okay, how do we know that calcium is so important for neurotransmitter release? Well, some of the earliest studies suggested if you just take calcium away altogether, then you don't get release any longer. We'll read about that in the Bernardo-Katz paper coming up very soon. But we can also demonstrate this experimentally. Imagine if we used a chemical called a calcium chelator. And the way that this works is it removes all of the calcium that's floating around inside the cell by binding it and sequestering it, or protecting it and not allowing it to bind in anything that it's floating around. If we do that with very high levels of calcium chelators, you see that we start to lose the postsynaptic excitatory postsynaptic potential. So here's a presynaptic action potential. Then we have these postsynaptic potentials. With less and less calcium, the signal gets slower and slower and then eventually degrades to almost nothing. Alternatively, we can use a different experimental setup where we have what's called a photocaged calcium molecule. Now, what's cool about this structure here, you don't have to know this structure or know what it's called, but this is what's called caged calcium. And what that means is that this calcium is trapped in this chemical structure. As soon as we shine light onto this structure, though, this little connection here with this nitrogen to a carbon is sensitive to light. So as soon as you shine light here, this breaks this chemical bond, and that allows calcium to diffuse freely out into the area around this chemical, where it can then bind and do all of its work. So imagine, if you will, we have some of this chemical. This is this nitrophin, which is this chemically caged calcium complex. And let's say that it's filled this presynaptic terminal here. Okay, so that's what this red is here in this section. Then we can shine light directly onto this, this molecule. That results in very rapid increases in calcium inside of this area. Again, because this bond is broken, calcium is allowed to freely diffuse out and then interact with these neurotransmitter vesicles that are found here in this presynaptic structure. What we're then doing is recording voltage from this postsynaptic structure here. And what you see is whenever you shine light, you get a big postsynaptic potential, or excitatory postsynaptic potential, suggesting that the shining of this light the enhancement of calcium is sufficient to cause an excitatory postsynaptic potential. We're going to talk more about these terms necessary and sufficient. Different types of experiments will allow you to make different argumentative cases for the nature of reality in biology and neuroscience. So what this first experiment is doing is demonstrating that calcium is necessary, because if you take it away, you lose the excitatory postsynaptic potential. This second experiment is showing that simply elevating calcium inside the presynaptic cell is sufficient to cause vesicle binding and neurotransmitter release. Okay, so the vesicles are going to eventually fuse via these membrane proteins. The vesicle is going to dock, and it's going to sit very close to the plasma membrane. But there's a complicated choreography that takes place between all of these proteins in order to pull the vesicle closer and closer to the membrane until it actually fuses. Often these are called snap-snare proteins, this entire collective complex, and it's comprised of synaptotagmin, which is going to be our calcium sensor. This is where calcium is actually going to bind, and this is the mechanism by which calcium is needed to cause fusion of the vesicle. There's also a protein called synaptobrevin, this MUNC18, syntaxin, and SNAP25. These things are all going to form a very sophisticated sort of twisty tie structure, if you will. Then they're going to wrap around one another. And as soon as calcium binds to synaptotagmin, that is going to cause this to twist even tighter, drawing these two membranes so close together that they are going to fuse, and neurotransmitters are going to spill out into the synaptic cleft. Okay. After calcium levels become low again, this is going to allow this thing to open a little bit further by relaxing this protein structure again. Okay, it requires some energy to untangle this web of proteins, and then we can start the whole process over again. Okay, interestingly, the membrane that is comprising these vesicles is the same type of membrane that comprises the plasma membrane. So, if you remember, one key element of any neuron is dictated by the amount of plasma membrane. And what's happening when we have vesicle fusion is you get more plasma membrane. And that key element is, of course, capacitance. So capacitance is a factor that is dependent on the amount of phospholipid bilayer that's available in a cell. So bigger cells have larger capacitance. When vesicles bind the membrane, what you see is there is actually more membrane, and that results in a change in capacitance. So in this experiment, these researchers were looking at the level of capacitance during a lot of exocytosis, or these binding events of these neurotransmitter vesicles. And what you see is they got step increases in capacitance every time one of these little vesicles bound to the membrane and became part of that membrane. Okay, we then need to recycle some of these things so that we don't have to continue making them, which would be very energy inefficient. So the membrane must be retained too. That's another good point. If the membrane gets to be too big, then the cell is going to start to lose its structure. It's very important that it maintains some structural integrity during this process. So there's a couple different things that can happen. Vesicles can completely bind with the membrane, and then this clathrin molecule can make this really cool looking complex structure and bring membrane back out of the presynaptic cell back into its cytosol here inside the presynaptic terminal. Another mechanism that can be employed to do this same sort of thing is the so-called kiss and run transient fusing of these vesicles to the outer membrane. Here you see this brief transient fusion that occurs, and then it doesn't fully bind and instead floats back away to maintain the similar amount of plasma membrane. Excuse me. Another thing that can happen is just bulk exocytosis, which again, if the cell terminal is just getting too big, the cell might need to maintain that by just taking up a whole bunch of this endocytosis. I was taught this as pinocytosis, which you might remember from biology as cell drinking, um, or these vesicles are formed with this extracellular solution in it. Okay, the last thing I want to mention today is this concept known as the tripartite synapse model. This is the three-part synaptic model. So instead of just having a presynaptic and a postsynaptic element, most of these synapses in the brain are surrounded by an astrocyte or an astrocytic process, which just means a little section of these astrocyte cells that kind of make this little contained chamber here to stop neurotransmitters from just freely spilling out all over the place. Astrocytes play a really important role in regulating excitability in the brain, not only through spatial buffering, which is a inward rectifying potassium channel leading to a reduction in extracellular potassium, but they also take up neurotransmitters like glutamate, which is the key excitatory neurotransmitter in the brain. What happens when this is taken up by these astrocytes is it gets converted into an inert form of this molecule that can then be cycled and re-entered back into a vesicle after it gets processed chemically to become active again. So through this process, astrocytes are master regulators of excitability by sequestering extra neurotransmitters and sequestering extra potassium. Okay, that's everything I have. Thank you for your attention. George, say bye-bye.
Bye bye.
Take care, everyone.